Natural Resources and Management at Texas Tech. And their names are Matthew Barnes and Nathan Gill. And so we're going to have two talks, one right after the other, and then questions at the end. So they have been here for a few months on this study abroad program where they bring a whole class of students from Texas to Spain to take some classes. And from what I gather, it's gone very well. And they're hoping to do this again. And so their lines of research fit very well with the lines of research of EBD, fire, water, temporary ponds, all that good stuff. And so this is a nice opportunity for us to get to know some people who may be coming back.
With you all. I'm really excited to talk about my research very briefly and then hand it over to Dr. Gill to talk about his research. Nos encanta, Sylvia. We have really enjoyed our time here. And so an average. My research sound exciting? So maybe I can find some collaborators and have a good excuse to come back. Forgive the fonts look So I do want to say that my background is as an aquatic ecologist. And I got my start in graduate school. Biological invasions, like the movement of bighead and silver carp, which were introduced in the United States, in the southern United States. They're incredible filter feeders and they do a good job. They were introduced to keep catfish farms clean by eating all the algae and plankton in those systems. However, they escaped the farms that they were in during flooding and started moving up the Mississippi River, which goes through the middle of the United States. And by the time I was doing my PhD, they were very close to, these are the Great Lakes in the north of Michigan, and they were very close to entering into the Great Lakes. And there was a lot of concern that these fish that reach sizes of around 45 to 50 kilograms as adults could cause a lot of trouble to the local ecosystem by eating all the algae and all the plankton. And so we were really interested in studying these fish. There's also, if you go to YouTube, there's a lot of videos. They have this really interesting defensive behavior. When they get scared, they launch themselves out of the water. And so this is behind a motorboat traveling up and down the river. The fish are jumping, and it's very funny, right? It's absolutely hysterical. Unless you're driving your boat down the river and a fish smacks into you. There's actually stories of people getting knocked out and falling in the water and drowning and dying by being hit by these fish, right? So a very important biological invasion concern. But this is in the middle of their range where they're abundant. At the leading edge of the range, they're not so abundant. They're hard to find when there's just one or two fish out there. But that's the time when we could actually take some management action and control them. Once they've gotten this large, it's going to be very difficult to get rid of these fish. They were so hard to find that we had to come up with other ways to look for them in the environment. And we said, you know what? These fish are swimming around and they're shedding genetic material into the environment in the form of sloughed skin cells and blood and other wastes. And so we can come along and collect that material from the environment and analyze the DNA that they shed into the environment, which we call environmental DNA, and use that to provide clues in the environment. And this sets up, in the United States, I'm not sure if there's a Spanish analog, but we have the TV show CSI, Crime Scene Investigators. So my lab is a crime scene investigation lab, only we're not looking for criminals at a crime scene, we're looking for invasive species or threatened and endangered species in the environment. And like I said, I'm an aquatic ecologist by training, and we have used these eDNA, these environmental DNA methods, to look for all kinds of species out in the environment. Other introduced non-native species like zebra mussels, threatened endangered species like the blue sucker or various mussels in the rivers of New Mexico and Texas, as well as other species of management concern like amphibians and wildlife pathogens like chytrid fungus or ranavirus. My PhD student works out in the deserts of Arizona and New Mexico looking at the spread of amphibian diseases using these genetic methods. So that's my background in aquatic ecology. What I want to talk about for just a couple of minutes about my current program is my emergence out of the aquatic environment and some of the work that we're doing with environmental DNA in terrestrial systems. And anyone who suffers from allergies knows that there are some species that release a lot of DNA into the terrestrial environment. And about a decade ago, about 10 years ago or so, one of my PhD students, Mark, showed up and said, my background is in terrestrial plant ecology. Could we use your DNA methods that you've been using to learn about mussels and fish and amphibians to study terrestrial plant communities? And I said, sure, let's give it a try. And that's what I want to talk to you about in the next couple of minutes here, is some of our work in the field of no longer are we taking water samples or even sediment samples or swabbing frogs. We have developed methods where we can sample the air around us and find the DNA of organisms in the environment. And we're going to start with plants, and we'll move on from there as much as I have time to do. We have this in Texas Tech. So Texas Tech is located in the panhandle of Texas. We have an experimental rangeland property. It's about 50 hectares or so of fairly native, ungrazed shortgrass prairie. And we set up these dust collection traps in a grid across that property. These traps are a triangle, a prism, with a hole in the front and a mesh screen or window screening on top. So the wind blows into the hole and up and out, and any of the dust or DNA that's being carried in the wind settles in the bottom of this trap. There's a tail on here, and this is on a pivot, so the hole is always facing into the wind. Okay? So we deployed these traps out on our rangeland property and sampled them. Mark said, well, how do you collect environmental DNA samples? And I said, well, I'm an aquatic ecologist. All I've ever done is collect water. So what we originally started doing was he would go out, he would pull those traps, and he would rinse them out and essentially turn them into a water sample. He would take that liter of clean water, rinse the trap out, and then he would take that water back to the lab. We would filter it, and then we would use standard DNA extraction methods to get all of the DNA that was floating around, what was in that dust, that we put into that water, and then eventually captured on our filter. And I'll show you in the next couple of minutes. We've done some species-specific approaches using PCR. We've also done some sequencing approaches where we no longer are asking, is one specific species present, but tell us about what can we learn about the entire community that's there. So a couple of early experiments. We wanted to understand how do the methods that we choose influence our results. And so here we are comparing different types of DNA extraction methods, a CTAB chloroform extraction, and then 2 commercially available kits. And on the y-axis here is the CT value, and this is analogous to how much DNA is present in the sample, except that there's supposed to be little DNA molecules here. Lower numbers mean more DNA, higher numbers mean less DNA. And the DNA that we're quantifying here is plant DNA in general. We're not looking for a specific species yet. The extraction method that we used gave us different amounts of DNA from the same. So methods matter. We also over here looked for 2 different plant genes. We looked at a nuclear plant gene and we looked at a chloroplast gene. Plants have 1 nuclear genome in each one of their cells. They have multiple chloroplasts in each cell. So we would expect to find more chloroplast DNA out in the environment. And indeed, because lower numbers here mean more DNA, when we looked for chlorophyll or chloroplast DNA, we found more of it in the same samples than the nuclear DNA. So this aligns a lot with what we have learned in aquatic systems, looking for animals. And so we thought we were on the right track here. Then we were thinking about this allergen, this pollen question again. Are environmental DNA from airborne sample methods, are they only useful for those plants that produce large amounts of pollen and have evolved to release large amounts of airborne environmental DNA? Or could this be useful for other species as well? And so we looked in our samples for blue grama. It's a grass, it's wind pollinated. It is, over evolutionary time, has designed itself to release a lot of airborne eDNA intentionally. We also looked for honey mesquite. This is an insect pollinated tree. It is not designed through evolution to release a lot of DNA into the air. It is, however, very if we're going to detect something, it's going to be this guy. We found both of them. So we were in our samples able to find a wind-pollinated species, but we were also able to find species that are not wind-pollinated. It gives us more confidence that this airborne eDNA method may really have some utility. Now we turn to the sequencing results really quick, and we tried to quantify the number of species that we identified in our airborne eDNA samples and compare that to a pretty comprehensive survey with transects and visual surveys that we did across that property. On the left is there's supposed to be a DNA molecule here. So in the blue, there are plants that we detected using DNA. In the red are plants that we only detected visually and we did not detect their DNA. And then there's a suite of shared species. For plants that, apologies, I know there's a beautiful plant collection here, and I apologize, I'm not a plant guy, so I'm going to very coarse kind of labels here. For the plants that have showy flowers, pretty flowers, we see a pretty even distribution. We had a fair number of species that we were able to detect that the DNA didn't detect, and a lot in common, but still some species that DNA detected that the visual surveys didn't. For the other category, and again I apologize to the grass scholars in the room here, but for the more boring species, the plainer species, we got a lot of unique detections with DNA methods, and very few that we found visually that didn't turn up in our DNA samples. And there's also four tree species on this property. We found evidence of all of them in our DNA, and there were no trees that we only detected visually that we didn't detect with DNA. Now, how did we miss a couple of tree species? It has to do with how our transects were randomly distributed across the landscape. This is a small property that is used in our plant identification classes, and our undergraduates go out every semester and identify, categorize every plant on the landscape. So we have a really excellent knowledge of all the species that are out there. But you could imagine going to your study site, or perhaps a new study site where you don't have the luxury of knowing every single tree that's out there, and maybe that one tree of heaven doesn't end up on your beautiful GIS randomly distributed transects. This gives us a little bit of hope that the eDNA methods might detect it. Okay, so then I want to, I think we've shown with a snapshot that the airborne eDNA methods are useful for learning about plant communities. So what? Can we learn anything ecological with these methods? And so we're going back out to this same landscape. We had an undergraduate class that was learning about invasive plant control. And they marched out there with chainsaws and chopped down a lot of advantage of this. We sampled the airborne eDNA before, during, and after this activity to see if we got any response in the amount of DNA that we quantified. And so let me try to orient you to what the graphs are going to look like. The graphs are going to have sampling dates on the y-axis, and on the x-axis is going to be the quantity of DNA that we detected. We were using a species-specific assay, and so the quantity is actually going to be an amount of DNA. So you'll see small numbers means a low amount of DNA. Numbers get bigger, we detected more DNA. We collected environmental DNA samples once and twice. Then our undergraduate group went out there, stomped out there with a bunch of chainsaws, chopped a bunch of trees down. We collected more DNA. They did their thing one more time, and we collected DNA one more time. The other thing I'll note, this is November 10, November 17. Either the Spanish or the United States is doing our dates backwards. We can talk about afterwards who does it right. But notice, a little bit of DNA. We had 3 different sites that we collected from. A little bit of DNA, a little bit of DNA. This is the end of the season. The trees are senescing. They come out there with their chainsaws. We get this massive plume of honey mesquite DNA in the air, seemingly in response to this disturbance on the landscape. And that happens over the time of their activities. We also quantified the DNA from blue grama, that wind-pollinated grass that wasn't a target of the study. However, you could imagine a class of 40 undergraduate students marching across the landscape with chainsaws is going to cause a lot of disturbance. And what we got to squint a little harder to see this one. We think early on we're seeing DNA decline because it's the end of the season, the above ground biomass is senescing, DNA is declining seasonally, but then we have that activity and everything jumps back up a little bit. We get at least, again, squint a little bit, but we get at least a little bit of signal of some activity on the landscape. So we think that eDNA is showing us seasonal and disturbance activities on the landscape. In the interest of time, I want to speculate just a little bit about whether the sky is the limit for this airborne eDNA methods. And in case there are any animal researchers in the crowd, I will point out that several groups out of Europe have begun applying these types of methods to detecting vertebrates and invertebrate DNA from dust and other things blowing in the wind. We saw that and said, well, that's a pretty good idea. And we've got a bunch of samples that we collected across the landscape for plant DNA. Let's take those samples and let's see if there was any vertebrate DNA in our samples. And so we quantified a vertebrate mitochondrial marker, and we found this is over time from June all the way until June of the following year, every 2 weeks or so over a year, we quantified and looked for detection of vertebrates. Just a couple of fun stories to see. This habitat has abundant rabbits and hares, and we got their DNA very frequently over the course of the year. Other species, like certain birds down here, we saw more seasonal. Lubbock exists in major migratory flyway, and so at certain times of the year we saw certain migratory bird DNA appear and then go away as they pass through. My other favorite on here, I'm an aquatic ecologist. Lubbock is in a very arid climate. We had essentially one major rainstorm over the course of the year. Right following that one major rainstorm, we got our only detection of the Texas toad, an amphibian that spends most of its life, sort of, at least in this condition, buried down in the mud. When we get a big rain event, that's its signal to come out and do its biological stuff. And we seem like we captured that in our airborne DNA, in the dust blowing through the air. Our detections in terms of the amount of alpha diversity, the number of species we detected at any given week, has some relation. It's messy, very low predictive scores, but seems to have some relation to rainfall. The more it rained, it maybe washed DNA out of the air. Also, there's probably some artifact that may have washed some DNA out of our traps as well. We got some collection issues there. Wind, Lubbock is a very windy place. Wind speed correlated positively with the amount of species that we detected. We think because it's stirring that DNA up. A lot more to learn here, but some interesting ecology to learn about as well. Mark and I were able to write a review paper. The field is growing rapidly and almost exponentially at this point. This is year and number of publications on airborne environmental DNA. I put this 2024 bar in blue to remind myself, to remind you. This paper was written in 2024, so this bar was incomplete, and indeed we continue to see this exponential growth in airborne environmental DNA detection. And as we move forward, we need to think about our foundational understanding. What is this material? Where does it come from? Where does it go? We need to think about how can this type of method be applied to what species and what systems. And we need to think about how can we synthesize, how can we use these methods to ask really exciting questions in ecology and evolution. And like I said, I'm really hoping to find some collaborators, so I have an excuse to come do some of that in Spain. And rather than take questions right now, I'm going to hand it off to Dr. Gill, and I think we'll do questions at the end. So, thank you.
All right. Well, thanks to Dr. Barnes for sharing that, and yeah, I'll try and make sure we've got time for questions here at the end, and those can be for him as well as me. So, thanks for pulling that up. I'm Nathan Gill, also from Texas Tech University, and my lab is the Landscape Ecology and Fire Lab. So I'm a geographer by training, and specifically I study the geography of fire. So spatial patterns of fire, ecological and social systems, and how they're related to those patterns of fire. Today I'm going to talk about forest structure, pyrogeography, or that geography of fire, and the conservation of a threatened species. I'm going to begin with a little story about one species in particular that maybe some of you have heard of before, the spotted owl, which is in the United States kind of a poster child of conservation science, and Basically, the spotted owl is this charismatic bird in the Pacific Northwest. It lives in wet, temperate forests up in Washington State and Oregon and Northern California, and was one of these early protected species under the Endangered Species Act that was very widely publicized and drew a lot of attention and kind of became this face of the conservation movement, and you know, kind of the reason that people were using to put forward ideas about conserving old growth forests in particular. The spotted owl relies on old growth forests with these snaggy trees that have been broken, large trees with high levels of canopy cover, and so, you know, it was basically put forward that in order to protect this species, we need to protect those oldest forests. And this did cause some conflict because a lot of people who live in that region of the United States rely on the timber industry, and it's how they make their livelihood. And so we had what is now affectionately known as the timber wars in the United States, where it was sort of conservation science of protecting these old growth forests, and these cute owls sort of pitted against the timber industry and the you know, extraction industries. And so it did get pretty contentious. This was when we had people start kind of chaining themselves to trees to try to protect you know, the forest from the big bad lumberjacks. But this has been sort of an evolving story. Of course, this kind of tension between that extractive industry and conservation has continued, but it's really kind of evolved as well. And I'm going to talk about how it's sort of changed with one subspecies of the spotted owl in particular. And that's the Mexican spotted owl, and that's the species that I'll be talking about with a study that I did on habitat for this species. So Mexican spotted owl is it's another spotted owl. It's a subspecies of that same northern spotted owl is the one that I had referred to that kind of launched this whole kind of debate and tension between extractive resource use and conservation. The northern spotted owl, as I mentioned, lives up in the northwestern United States. The Mexican spotted owl is, of course, very similar. It's a subspecies and relies on a lot of the same sort of forest structure habitat requirements, but it lives much further south, down in Mexico and in the southwestern United States, even a little bit into Texas, and this region of southern New Mexico is where I'll be presenting results from. But it's a little bit different because of this different range of the United States that it lives in. And the primary threat that was described for the threat to this species when it was listed under the Endangered Species Act was fire, and stand replacing fire in particular. So rather than you know, timber industry as kind of the primary threat down in the southwestern United States, we don't have as much. We still do have some you know, timber extraction going on, but fire is really a big part of the disturbance ecology in those forests. And not only is this seen as a threat to the destruction of the habitat of this spotted owl subspecies, but this is also a region where we are expected through climate forecasts and what we think is going on with sort of the accumulation of fuels in this part of the world. We're expecting to see a 13-fold increase of the amount of fire that is happening in these landscapes. So we're already seeing fire destroy a lot of the conditions for nesting for this species, and we're only expecting to see more in the future. So what could be one possible way that we could help address this threat to this species? Funny enough, one idea that was put forward was, hey, we need to go cut down the forest and then it won't burn. And so we kind of have this whole situation where the story has kind of flipped on its head, and now there's really, frankly, a lot of confusion as to what is the right way to protect this species, what habitat does it need, and how can we possibly manage the landscape in the face of climate change and other human activities. What's kind of the right way to protect the habitat for this species? And so we started realizing that we really need to have a better understanding of the relationship between the Mexican spotted owl and fire. Clearly, given its range in these southwestern U.S. forests, the Mexican spotted owl evolved alongside fire and very fire-adapted forests. So we know that this, fire is not a new threat to this species if we're just speaking in very general terms. But there have been, I guess I should add also, people have observed these Mexican spotted owls as well as the California spotted owl continuing to nest and use habitat in forests that have burned at high severity. So it is possible, although somewhat rare. But also the Mexican spotted owl has some parts of its range where there is no forest, and they actually just dwell in canyon systems. And so there's kind of this whole question about, well, how much do they really need those same old growth forest, thick canopy conditions to be able to thrive? And again, we can look at sort of the map of fire patterns that we see. So this is just showing all of these different colors to show us the fire regime, and that's kind of a funny term that we use to just describe the repeated pattern of fire. So how frequently do we see fire that keeps happening in an ecosystem, and how severe does that fire tend to burn? And we can kind of categorize that to have different types of fire. And we see Mexican spotted owls down in this portion of these kind of green and blue shades that are a little hard to see on this map. But that's basically understory fires, so low severity fires that don't spread to the canopy, that repeat every 0 to 34 years, maybe some that are a shorter interval of 0 to 10 years. And then, kind of in these lower elevation ranges, some mixed severity fire, maybe a little bit getting into the canopy or some patchy higher severity areas, but still at an interval of 0 to 34 years compared to hundreds of years that we might see in places that are a lot wetter or higher elevation. And so I focused this study on this portion of southern New Mexico here, not far from Lubbock. And it's the Sacramento Mountains, which are a sky island ecosystem. And so what a sky island system means is that it's sort of these mountains in a sea of desert, right? So we have this more precipitation, even snow and true forest growing at these higher elevations, but they're really just these kind of islands surrounded by much drier environments and grasslands. And that creates an interesting situation with the fire regimes, because we can have fires that will start down in this kind of frequent fire, low severity grassland system or shrubland system that could spread into the true forest that might burn at higher severity, might be more adapted to burn at kind of longer intervals than what we see in the more arid environments. So again, just to give kind of some specific snapshot of the kind of disturbance history of these fire regimes in the Sacramento Mountains in particular, we've got a couple of different forest types. Many are dominated by ponderosa pine, and those will usually burn at low severity every 3 to 10 or 11 years. Maybe at higher elevation than that, as we kind of get further upslope. We've got mixed conifer forest that might be just a slightly longer interval between fires. And this is often driven by these climate oscillations and also heavily influenced by human land use, right? Humans will be often the cause of ignitions that start these fires, but also if people are grazing or cutting trees or clearing the land, that also affects how much fuel is available to burn, which in turn feeds back to these patterns of fire, these fire regimes. So I focused on these forests in Lincoln National Forest inside the Sacramento Mountains. So this is public Forest Service land in New Mexico. The boundaries are shown here, and then this is showing that mixed conifer forest, so kind of that mid-elevation forest type with, you know, those pretty short, you know, 0 to 15-year fire return intervals is what we think is kind of the historical norm in these forests. We have 2 different districts in this forest that we collected data from, the Sacramento Ranger District here in the south and the Smoky Bear Ranger District in the north. This is actually where Smoky Bear came from. There was a bear that was rescued, you know, 100 years ago from a wildfire, and he was found in that district of the Forest Service. So that's how it gets its name. Then in the middle here, you know, dividing, I guess, these 2 districts of Forest Service land is a Mescalero Apache Indian reservation. And so the Apache who lived here for obviously much longer than the Forest Service has been around, used fire as a tool to help manage the land. But after Western colonization and the displacement of those peoples, a lot of that fire management and use of fire on the ground was gone as well. And so they continue to use fire as a tool. The federal government of the United States has started to also realize some of the benefits of using fire as a tool. So in modern times, inside the Forest Service, we see some prescribed fire, but really it's only now that we're recognizing that we need more of that to kind of maintain the natural fire regimes that have been part of this system. I want to circle back to the spotted owl, though, and just paint this sort of unclear picture of, okay, so what does this species need? We've got a couple of papers that kind of came out after this, you know, listing as the species being endangered, and actually it wasn't until a decade or 2 later that they really identified high severity fire as the primary threat to this species, and that's where a lot of these questions started to arise. So we have this review from Juan et al. That found that a lot of studies on the Mexican spotted owl were focused on very short-term effects, and very few of them even bothered to quantify whether the type of fire that we were seeing was high, medium, or low, and it kind of was just a dichotomy of fire or not fire. We had other studies that found that, hey, owls were surviving, reproducing, mating, remaining in place after fires come through and continuing to have site fidelity at their nests. Occupancy and reproduction in burned areas was maybe slightly lower than unburned areas, but people started paying attention to some of the different fire severities and noticing that, hey, patterns might be different here. And then interestingly, Gainey et al. In 2014 found that owls actually foraged more in areas that had been burned, and they were finding more things to eat in that forest that had been burned and kind of cleared and made more visible. So even if fire might be bad for the nesting habitat conditions for Mexican spotted owls, it actually might be doing a service for the species by making it more possible for them to find food. And interestingly, none of these studies up to this point, and at the time that we did our study, no study on the Mexican spotted owl had examined any patterns of fire more than 6 years after fire. So that led us to sort of question, well, you know, we know the effects from fires can last a lot longer than just 6 years, and most of these were only 1 or 2 years after fire. So we wanted to get at that a little. So the questions that we wanted to ask are, how does forest structure vary in stands that have different histories of fire severity and time since fire, and look a lot longer back? So we actually went 100 years back. And which patterns of fire are most conducive to Mexican spotted owl nesting habitat? And again, that nesting habitat is kind of seen as the bottleneck, or the thing that's being threatened by this fire that's happening. So we already knew from prior studies that there are certain conditions, things that we can go out and measure in the forest that are especially important for nesting habitat for these owls. And so this table is just showing kind of what some of those thresholds are for the kind of forest structure that you need to maintain good habitat for these owls. And I'll show some graphs later that will basically have a purple line on them to show where that threshold is. We went out to 113 plots kind of scattered across those forested areas that I showed you, set up these circular plots to collect basically these same variables, measuring these crucial aspects of the forest structure. And we did that across areas that had different fire severities and time since fire. And actually we knew how long it had been since those areas had burned, but we used tree rings to try to help tell us how severe that fire was. And we basically confirmed the date of the fire by figuring out when trees established. And then we looked at, hey, how many trees are still around that were there before the date of this fire? And that helped us get at least a loose idea on if it was a high, medium, or low severity fire. A higher severity fire is going to kill more trees, meaning there would be fewer survivors. So yeah, we ended up with kind of a good stratification across some very coarse, just kind of 3 levels of fire severity. And then we had fires that had burned 100 years ago, about 60 or 70 years ago, 20 to 30 years ago, and in the last decade. We ran a Permanova test on these structural attributes to figure out if there were differences in these different kind of fire history classes. And here's kind of a picture of what these classes looked like. So you can see fire that happened longer ago and high, medium, low severity fire. So we kind of get this grid of like how much vegetation is still there on the landscape. And it's obviously the longer we go back in time, the kind of harder it is to tell some of those differences, but you can still see them, and they're there, and we can kind of quantify them when we measure that forest structure. All right, so canopy cover is considered to be kind of the most important factor for quality nesting habitat for Mexican spotted owls. So that's the one I'll kind of show and focus on here, and then I'll go through quickly a couple of others, but they're all telling a pretty similar story here. So I'll kind of walk you through this one, but on the y-axis here we've got the percent canopy closure. So the higher this is, the kind of more canopy we have, which again is good for owl nesting. We got 3 different colors on most of these frames here, showing yellow for low severity fire, orange for moderate severity fire, and red for high severity fire. And then the different frames are the different time periods for when the fire burned. So these are the most recent fires, and as we move this way, we get to the oldest fires. And then the blue plot here is showing the control, or the areas where we went out that had no record of fire for over 100 or even 200 years, and compared that control group against what we were seeing in the burned areas. And so really the trends that we kind of see consistently across not just canopy closure, but some of these other metrics are that we've got this pattern of, okay, canopy closure is really low right after high severity fire. I mean, we would expect that, but it does step up with fire severity. And also, as we look at time since fire, and especially in that highest severity class, the more time we have after fire, the more that crucial habitat condition for owl nesting is recovering. And how long does it take? We were kind of trying to get at that. And basically for canopy cover, we saw that, hey, after 100 years, that forest had recovered to a point where it was above that threshold. And even, with an average canopy cover that was on par with our control stands. So we basically concluded from that after about 100 years, even a higher severity fire was kind of recovered for the sake of the Mexican spotted owl for canopy closure. And so, yeah, comparing kind of that control with the high severity fire is another thing we kind of want to look at. Here we have just other aspects of forest structure that I am not going to, for the sake of time, dwell too long on, but we see a lot of the same patterns where high severity fire really kind of eliminated those conditions, but over time or with lower severity, we're seeing kind of more recovery. So density of large trees, right? There are about 5 or 6 of these. I want to get to kind of my takeaway messages though. So for the Mexican spotted owl, obviously high severity fire has the most deleterious effects for nesting habitat conditions. We expected that, but we think we found some valuable kind of specifics as to time to recovery after fire, and if there are some differences with different severity classes. With more fire, we do expect to see more high severity fire, more lost, structure for owl nesting. But stands that burned at medium severity generally recovered more quickly than the stands that burned at high severity. And of course, low severity fires sometimes recovered very quickly. And by some metrics, stands that burned at high severity more than 100 years ago were actually even more suitable than some of the low severity fires or even many of the control stands that we measured. So that kind of suggests that, hey, it's good to have fire and then allow some time for it to recover and kind of create that structural diversity. And when we bring that into ideas like the Gainey study that I referenced, if owls need some conditions for nesting, but other conditions might be favorable for foraging, we can see how, okay, it makes sense that this species evolved with fire and maybe kind of this patchiness of different types of fire and time since fire. And so what does this have to do with kind of a bigger picture? If I don't just care about owls, but maybe I care about a European lynx or another species, right? I think for any species or even communities of species that we are interested in or that we have kind of a vested interest in, we need to ask ourselves not just what the disturbance history is in terms of disturbances being bad or good, or if fire is bad or good for that species, but, and I think that ecologists have generally come to understand that pretty well. But another way to look at it is not just, okay, we can get over this idea that there's a dichotomy between fire adapted or fire prone, right? That's one thing. I think a lot of ecologists now recognize that we maybe at least need to pay attention to that fire regime, right? Like, hey, maybe there's a certain interval or severity level that's most favorable for this species. But I would posit that we need to even go kind of one step farther and say that they're not just adapted to even one fire regime or one interval at which fire recurs, but patterns of pyrogeography, and that they actually need that mosaic. They use different types of habitat for different resources and different needs, and they, as we have time since fire and fire burning at different severities and in different spatial patterns, that allows us to kind of provide for those different needs for these species. And that's truly what these species are adapted to and have evolved alongside, is that diversity of fire and these spatial patterns of different fire. So I suggest that we need to kind of pay a little more attention to that higher level of differences in patterns of fire and what's present as a mosaic on the landscape. And with that, I'll wrap it up. I think I'm about out of time, but thank you for your attention. I do want to just thank the field crew who helped collect these data back in 2020 and 2021, and especially Tara Durborough, who was a grad student of mine who did a master's thesis related to this work. And so special thanks to them. And I'd like to turn the time over now to you all for questions for me or for Dr. Barnes. Thanks.
Okay, I have a question about the airborne DNA. I would like to ask you, how big are the amplitudes you are able to get? And how much is the sequencing depth you need to reach the asymptote in the condition group?
Okay.
The second is not so easy. Maybe to answer right now.
Okay, those are, the sequencing depth is a great question, and I'd have to look at I'd have to look at the old sequences to tell you that, and probably have to get my PhD student on the phone. In all honesty, so I'm going to punt on that one. What's the next one? That's
What was about the like amplicon size?
Oh, the amplicon size. So those for eDNA, we tend to favor shorter amplicons, right? The DNA that's out there in the environment is slowly dying. It's degrading. And so anything that I showed you here today is in 200 to 300 base pair range. If we're going species specific, then we can go even shorter. So we would probably, for the blue grama and the honey mesquite, those were probably closer to 100 bases in size. Because if you look for that, we're getting better at this. People finding and assembling larger genomes from the environment, and actually some people have really started. Kara Andres in the Great Lakes, for example, done some really awesome work sequencing whole mitogenomes from environmental samples and doing some really cool population genetic work. But anything that I've shown you, we're looking at really short sizes, 100 to a couple hundred.
Okay, and did you try or are you planning to try to get like the same information from insects? Like you go like frogs, but are you trying with insects or arthropods?
Yes, yeah. So we have done invertebrates as well. We actually, one study that I wasn't able to show you due to time, we collected samples in a 200-meter meteorological weather tower up and down the tower and looked for, we quantified and we sequenced plants, vertebrates using 2 different mitochondrial markers and invertebrates up and down that tower. And found some really cool changes over time. One thing that we're dealing with right now, my student set his samplers up and down the tower, had this brilliant, every couple of weeks we were going to go out and sample. He deployed them in March of 2020, and we were able to we collected them like a week later, and then we collected them again like 120 days later, and the sampling, because of COVID, just looks like we had no idea what we were doing, and so we're trying to make some sense of that. But yes, we can do vertebrates, invertebrates, fungi. We did fungi as well, plants, and there doesn't seem to be any shortage. Right now, there's a moratorium. The eDNA community, we've agreed not to do any people DNA, but unfortunately, there's probably a lot of that in our samples too, and we'll deal with the ethical repercussions of that in the future, for sure.
You want me to wait, or? Okay. I have another question for you.
Of course.
So, are there groups of organisms for which you have good survey data to have estimates of amount of, like, number of individuals, etc.? So, because ideally, you could try and do some surveys through environmental DNA, but the translation from like quantification is, I guess, is complicated, and so not only individual abundance, but actually like activity patterns, I guess, would matter. So not only how many individuals there are, but how active they are, or what kind of stage of their life stage.
Yeah,
life cycle
yeah,
they are,
yeah. So my passion, my research lab, I say we're an ecology of eDNA lab. There's so much that happens in terms of the origin, where this stuff comes from, how it's produced, its state, what its condition is in the environment, its transport, how it moves around, and its ultimate fate. How quickly does it degrade? That stuff, a lot of ecology happens after the DNA is shed from the organism, but before we scoop it up. And the more we learn about that, the more useful the tool will be. We know, I'll fall back on aquatic systems for now. We know from studies in fish, for example, younger growing fish give off a lot more DNA per biomass than larger fish. Larger fish, though, have more biomass, and so they give off more DNA in total. And now compound that. That's an experiment from little fish tanks. Now think about that at a community level, right? So the answer to your question about can we relate the amount of DNA to the number of organisms, the amount of biomass, the answer is yes, but with very large error bars. And I can say and generally just relatively speaking. So I can say there's a lot of blue grama and there's less honey mesquite. And that's about as scientific as I can get right now. We can do comparisons like that. I think that's where we're headed. But we need a lot more calibration experiments and comparison, those side-by-side kind of experiments with surveys, trapping, and the eDNA sampling to say how do these relate.
All right, so that's why I was wondering if there are specific groups like rabbits. So is there like a survey of traps? So would you have any pairing time series of abundance, like number of individuals for some groups that you could try and calibrate?
Yeah, I don't have those data here, but again, because the property that we're working on is, in addition to plant surveys, we've got years of small mammal trapping, for example, and we might be able to get at some of that. So maybe I'll come back next year and I'll tell you about our brilliant result. Yeah.
I was wondering about the scale of the fire in your dataset. So it's related to what you finished with.
Yes.
Yeah, so I was wondering about, in your historical data that you've used, what sort of scale those fires happen, and including in relation, spatial scale, I mean.
Yeah.
In relation to territory size of the owl.
Yeah.
And whether those would be highly territorial and faithful to their territory, or whether they are, moving.
Yeah, great question. In terms of, I don't remember off the top of my head kind of the exact scale or size of any particular fires, but I would say as large as like maybe 100 or a few hundred square kilometers. And so most of the large wildfires that we have out there will be larger than the territory size of the Mexican spotted owl, and assuming that it makes it not suitable for them, would displace them, and we've seen some of that actually with their nesting patterns. But there are also many smaller fires that kind of happen and don't get a lot of attention and just burn a small area and then go out. And so there is kind of a mix. But yeah, many of the larger ones are large enough to completely encompass the territory of many pairs of owls.
And so would the higher severity fire usually be, also on a larger?
Yes, that's a good point. That's true. The higher severity fire would typically be the ones that are affecting a greater extent. Yeah. Good question. Good point. Yes.
Thank you very much for your talks. They were very interesting. I would like to ask about the prescribed fire practices. I don't know if they are already implemented in the area you are studying, and I would like to know how they are performed. Like they are just for the under canopy cover, or they are also take down the trees or something like that?
Yeah, great question. So I was mostly working in, because of where Mexican spotted owls tend to be, this study kind of took place more in the mixed conifer forest that's a little bit higher elevation, a little bit adapted more to some higher severity fire, kind of in the natural regime. But down, especially in the ponderosa pine forest that's just below that, that's where we see a lot of the prescribed fire that is lower severity, just surface fire, when we can do it on a day in the spring. Maybe there's even snow on the ground, so it's kind of a very mild fire that we use. And it's actually interesting, we've started to see more of that from the Forest Service and partners with the Forest Service in southern New Mexico. But they had a historically large wildfire in New Mexico a few years ago that started from an escaped prescribed fire and burned several people's homes and was a catastrophe, truly. And so after that, the Forest Service for a year said no more prescribed fire. And now they've opened it back up, but it's been a very politically fraught conversation, and it's very hard to help people understand that we want to protect your home from fire, so we're going to burn, right? It just doesn't make sense for people. So it's it's a really interesting kind of part of the story. But when we understand the fire ecology, we know we need more fire on the ground. It would be helpful to help prevent the bad fire or the high severity fire that's not typical of this system.
Hi, so early in your talk you mentioned that you were able to identify insect pollinated plants in the environmental DNA, but I guess you were referring to non-functional pollen in any case, or maybe other tissues, right?
Non-functional, say that one more time?
So I guess that you were referring that this DNA is coming from other tissues, but pollen or functional pollen.
Good question. I mean, I absolutely think it could have been pollen, right? We, just because these plants aren't wind pollinated doesn't mean in our very windy environment where we have lots of that tree in particular that there's not some pollen blowing in the wind. We haven't analyzed. I really can't tell you what it is that we're collecting when we collect this stuff. Anecdotally, we in those traps find a lot of dust that probably is pollen, but we do find plant fragments, little pieces of leaf, little debris, and certainly that makes up some component of what we find when we find what we collect when we collect airborne eDNA. But we don't necessarily have a good grasp on exactly what the composition of that stuff is. Does that make sense?
Yeah, I'm just wondering if, to what extent this airborne pollen from non-pollinated plants can have an impact in their pollination.
Mm-hmm. Yeah, and we probably were collecting some pollinators as well, too, right? Like they're very well, we're definitely, we got a fair number of spiders in our traps that just were living in there. I would feel very uncomfortable trying to say that we caught spider DNA blowing through the air, because most of our traps had spiders in them, and certainly some of our samples had a whole spider, get washed into the DNA extraction. So, yeah, still a lot to learn about what that material is, and it's going to be really important.
Thanks, Miran. Very cool stuff.
Okay, so thank you for your talk. And
thank you, all of you.
Muchas gracias.